Hi everyone, thank you very much. I'm Olivia. I'm a statistical scientist at Plant and Food Research in New Zealand, and today I'm going to talk about a little R package that I've been working on about visualization. So just as a bit of context, during my PhD, I worked on autotetraploid potato, and specifically I was looking at the genetic components that are involved in tuber bruising. And so for this work, I got some nice genomic data, phenotypic data. So naturally, I went for GWAS analysis that I did using the GWASpoly package. But I also was lucky and I got some transcriptomic data, some gene expression. So I did a differential expression to find out what were the genes that were differentially expressed between low bruising tubers and high bruising tubers. And so what I ended up with was I had some GWAS results that show me the markers of interest. As we've seen a lot during this talk, we typically represent them with Manhattan plots, right? Like we can see here. And I had differentially expressed genes, and typically we represent them with a volcano plot, which doesn't tell us much apart from we have differentially expressed genes. And on top of that, I had some genes of interest. So I'm calling them candidate genes, but it's really like when you're doing a literature search and you find out, oh, there are some genes that we know from previous experiments are potentially involved in tuber bruising, or from biology, it makes sense that they would be involved in tuber bruising. So I had these three types of information, but I really love plots. And I really wanted to have a visualization where I could summarize all of this information into one graph. I don't want boring tables. I don't want 3 different plots. I just want one nice graph that summarizes everything. So that's where we came up with this idea of the HIDECAN plot. And if anyone has a better name suggestion, I take it. So the idea behind this HIDECAN plot is that I want to show each chromosome as a little box, and I want to actually represent genomic positions. So in this HIDECAN plot, the x-axis that you see here is the genomic position on each of the chromosomes. And now the way that I'm going to represent this different information is I'm going to add tracks or layer in my plots on the y-axis. So starting with the GWAS results, what you do typically in a GWAS analysis, you have your marker scores, you pick a significance threshold, and anything above the significance threshold are your markers of interest. So I can take these markers of interest and I can show where they are located on the different chromosomes. That's going to be one track in my HIDECAN plot. I can do the same thing for differential expression. So typically in differential expression, you also pick a significance threshold on the p-value or the score of the genes, but you also pick a threshold on the log2 fold change to have genes that have a really different expression between the two conditions, right? So with these two thresholds, then I can select my genes of interest. And again, I can represent their location on the HIDECAN plots. And then finally, if I have candidate genes or like my list of genes from the literature, anything I'm interested in, I can also represent their position on another track, tracking the HIDECAN plots. Now, what that gives me is that now I have a representation of all my chromosomes, and I can really easily identify the genomic regions where I have information of association between genotype and phenotype using different layers of information. Okay, so that's what I was wanting to represent. So last year, when I gave a talk at the Tools for Polyploid workshop, I showed all my potato data. And one of the questions that I got was, how did you create this HIDECAN plot? And at the time, it was just an R script that I wrote for this project in particular. So my supervisor suggested that I turn that into an R package because I thought it could be useful for other data sets, right? So that's what I'm going to briefly show today. Very originally named the HIDECAN package. If you are interested in the HIDECAN package, it's publicly available on the Plant and Food Research GitHub. So you can just type HIDECAN GitHub and hopefully you will find it. It just got public and we're working on adding a GitHub page that has all of the documentation. So hopefully if you are really keen to see how it works, there's going to be documentation in the next weeks coming up in the GitHub repo. So how does the package work? Well, it needs some input data. And in this case, the input data, they're going to be data frames or table of the GWAS results, differential expression results, and list of candidate genes. When I'm talking about GWAS or differential expression results, I'm talking about all the results. So the marker score for all of the markers that you tested, not only the significant one. Technically, when talking about the R scripting itself, you need certain columns to be present in your input data frame, stuff like chromosome, position, score, etc. I won't go into detail, but there will be more information in the documentation. And in case you want to get started playing with the package, but you don't really have data that is in the right format, in the HIDECAN package, we have a function called getExampleData that will give you example data. What it looks like is something like that. So the example GWAS data, you have a data frame. Each row is a marker. So you have the ID of the marker. We don't really care about that. More importantly, you have the chromosome of the marker, its position in base pairs, and the score. Differential expression results are a bit more complicated in the sense that you also need to have the log to fold change of the differential expression analysis, etc. For candidate genes, you need to give a name because they are going to be labeled in the graph. That's really useful if you want to reference a certain gene that you know is involved in whatever biological pathway, etc. So the main function of the HIDECAN package is called HIDECAN plot. The first three arguments are just going to be the data frames that I just talked about. So you give the data frame of GWAS results, differential expression results, etc. And you also want to specify the threshold of significance. So above what score is a marker considered significant? Same thing for the differential expression. So you give a score for the, a threshold for the score and the log to fold change. And that's basically all that you need. And on top of that, you have a number of optional parameters that basically define what your plot looks like. Here, I just picked a random example so that it nicely fits the slides. So the output of this command will look like that. It's the output. So you have your HIDECAN plot where each of the box is one of the chromosomes. On the x-axis, you have the genomic position, and on the y-axis, you have these tracks. And you can see genomic region, okay, here on chromosome 3, I have a bunch of differentially expressed genes, then I have a GWAS peak not too far away, etc. That's the typical HIDECAN plot. Now, maybe some of you are thinking, okay, it's cool, but I don't have differential expression results. Well, it's fine. You can also use the HIDECAN plot if you only have some of this information. So let's say you just have GWAS results, and you also have some genes that you are interested in. You can use the HIDECAN plot function as well, just giving GWAS information, GWAS information, your candidate genes. You can also get a HIDECAN plot. You're going to have these two tracks corresponding to the information that you put in. Maybe more interestingly, if you have GWAS results for more than one trait, I know that a lot of us do, so we collect a number of traits, we do the GWAS analysis for several traits. So that's an example here. I have two data frames, GWAS results for two different traits. What you can do is you can give them as a list in the HIDECAN plot function. So rather than giving one data frame, you give a list of data frames. You can name your list. So that's the result for trait 1, that's the results for trait 2, and you're going to get a HIDECAN plot that has two tracks, one for the GWAS result for trait 1, one for the results for trait 2. And the names that you can see here correspond to the names that you gave in the list. If you don't give name, it's just going to tell you GWAS 1, GWAS 2. Okay, so that was the generic overview. Now the problem, not the problem, but if you want to use HIDECAN as I just showed, you need to take your GWAS results and put into, okay, I need to have a data frame with this chromosome column, etc., etc. Now, when I was working during my PhD, I was working with GWASpoly. And the really cool thing is I used GWASpoly to analyze different traits at once. And also I wanted to test different genetic models. So the output that I got was a lot of Manhattan plots. And I didn't want to compare all my Manhattan plots. So I wrote a little helper function that can directly take the output of GWASpoly and create a HIDECAN plot. And the reason for that is once I got all of my GWAS analysis results for different traits and different genetic models, I wanted to be able to quickly compare them and see what are the significant results if I use different genetic models or between different traits. So I'm going to show here an example that is using the original example dataset from the GWASpoly paper. I'm just showing you, let's imagine that I get the data and I run the analysis for 3 different traits and using different genetic models as well. So I've added this, this example dataset into the HIDECAN package, just if you want to run an example. So the output object from GWASpoly is an object of the class GWASpoly.fresh, which is the output of the set threshold function. So basically, it's an object that gives you for each marker their GWAS score for each trait and also for each genetic model. So trait times genetic model. And I wrote helper function that takes this output. So it's called HIDECAN plot from GWASpoly. And the only thing that you need to give to this function is the output from this set threshold function from GWASpoly. I've, just for the sake of the example, I've also added one of the optional parameters. So these optional parameters are also available in the base function, if you want. It's called remove empty chrome, just that if you have 20 chromosomes, but you only have 2 significant markers, you don't want a nice plot completely empty. So instead, it only shows the chromosome in which you have some significant marker or genes, etc. And you get something like that. So you can see the peaks or the significant markers for each GWAS analysis. That means GWAS analysis for each trait and genetic model. And that can allow you to do things like, okay, I looked at 3 different traits and I can see that there is a genomic region here on chromosome 10 for which I have significant marker for tuber I and tuber shape using either the general or additive model. But I can't, this region is not significant when I look at one of the simplex dominant models. So it's just a quick way of visualizing rather than looking at the Manhattan plot or like getting a lot of tables and comparing. I just quickly visualize the interesting regions. Now it's a bit hard to read actually. So if you want to focus only on some traits or some genetic models, you can just add, there's parameters for that. So you say, okay, I want my plot to focus on the tuber eye depth and tuber shape traits and only show the results for the general model. And so you get a simpler plot, so you can focus, zoom in, etc. Lastly, just because it's fun to do, if you don't really want to write a R script and you're more comfortable with clicking on, using an app and clicking to set the different parameters, I wrote a little shiny app that you can see working here, where you can input your data from CSV file. And as you're inputting the data, the plot will be created. And then you can select the significance threshold using sliders, which was actually a question of interest. Like if you allow for more significant differentially expressed genes, are they going to be located randomly or are they going to be located in specific regions? That can be interesting. You have these other optional parameters, like you want to add a title to your plot. So here you see I'm adding a title. I'm going to change the position of the legend, because why not? Almost there. And once you're done, then you can download the results. So let's say you want to do a presentation or you want to use it for a publication. Then you can decide if you want to save it as a PNG file or a PDF, and then you download. We're almost there. And you get the resulting plot. So I think I was really fast, but I think, yeah. So far I've shown the example where you can use the output from GWASpoly to create a HIDECAN plot. But I've seen a lot of tools that were developed that I think would be cool to be able to visualize with the HIDECAN plot. So I'm really open to any suggestions, or if you think there are things that could be improved in the plot, or packages that you would like to be able to convert into, like the results into HIDECAN plots, feel free to contact me. I would be really happy to add that. And we're working on a paper, a small paper to publish that. And so once again, if you are interested, the package is available on GitHub. The link is here, and the documentation will be there shortly. Thank you very much for your attention.